they'll be able to relate them. So please don't hesitate to reach out. So I'm really so happy to be here today to talk to you about a topic that's near and dear to my heart, which is aligning representations in brains and machines. And to kind of step back, I wanted to motivate a little bit why, I care a lot about this topic, but why I think you might be interested in it as well. So let's say that you're a neuroscientist and you're working with human data. And maybe you've thought a little bit already about the problem of comparing across individuals. And the idea so far is that, okay, yes, if I want to move beyond case studies, I need to have some way to map between neural activity patterns in participant 1 and participant 2. But the way that we commonly do that as a field in neuroscience is to align to some known anatomical template. The problem is that we've known basically as long as the field of cognitive neuroscience has existed that this doesn't totally solve our problem for us, right, in terms of making a mapping. So for example, this is a really nice illustration of one particular brain region, though there are many others we could call out, known as Heschel's gyrus. And within Heschel's gyrus, you see that depending on the particular individual you're looking at, this can present as a single gyrus, hence the name, or it can actually present as a completely duplicated gyrus. And it's not clear how to establish a mapping between these two different kinds of anatomy, right? There's no consensus. You wouldn't necessarily say, okay, everyone gets a half gyrus, right? So how could we make comparisons here? And maybe you say, okay, that sounds like it's a problem for humans because we're looking at these really high-level spatial scales. But if we go to a really fine spatial scale, or maybe if we move to a different model system with a different kind of anatomy, maybe that problem goes away. Unfortunately, systems neuroscience, neuroscience more broadly, has really seen that moving to higher spatial resolutions, even single neuron resolutions, or to animal models, does not resolve these challenges of how to make inferences across individuals. And even actually they've exacerbated the problem, because now when we have access to this really high resolution data within individuals, we see that, if you're doing invasive recordings with implant degradation, or even in non-invasive recordings, changes in neuronal plasticity and representational drift can yield substantial within individual changes and make it really complicated to compare the same individual's recordings over time. So here's an example just to kind of illustrate what I mean. So this is from Laura Driscoll and colleagues, but summarized in this really nice opinion piece by Rule and colleagues. And basically what they're showing is this is calcium imaging data from the posterior parietal cortex of a mouse while he navigates a T maze. And so what you can see is that here are the calcium imaging signals from these, a variety of posterior parietal cortex neurons. And if you look at Q offset and you sort by day 1's activity, you see that the mean activity in day 1 displays this really typical sinusoidal pattern that we expect. But then if you keep the cell sorting from day 1 and look at day 10 or day 20, it seems as though the effect has gone away, right? It seems as though the neurons are no longer representing the same pattern. But in fact, what we see is that if we resort by day 10 and day 20, that the pattern is persisting over time. The population activity is still encoding for the same concept in this T maze. It's just that the specific neurons that are being recruited have changed. And so if we create a decoder, for example, that relies on these exact neurons, it's going to start failing over time, even though the population activity is persistent, right? And so this makes it really complicated to do things like create brain-machine interfaces. And so maybe you say, all right, I believe you now. I could see that this would be a problem in humans. I could buy that this would be a problem, in neuroscience quite broadly, but this seems like it's all a biology issue. If I'm in silico, this probably isn't my problem. Unfortunately, in fact, it does persist even in silico. So if we're looking at artificial neural network instances, we see that convolutional and recurrent networks both show individual differences in learned representations. And importantly, this is independent of the classification accuracy that they're able to achieve. So even for near identical classification accuracies, you can see these individual differences, and these differences are persistent. So they're occurring even with the exact same architecture and with the same training set of data. This is only the initialization differing. So this is a really nice figure from Merrier colleagues kind of demonstrating the problem. Here we have a convolutional neural network, and we've taken multidimensional scaling, just the first 2 dimensions. And if we look at multiple instantiations of the same architecture of this CNN on the same training data, and we look over time across the layers, what we see is that in the initial layers, the representations are relatively conserved across the different instances, across the different individual networks. But as we move through the layers, we see more and more individual differences in terms of how each layer has representations that are being orchestrated by the hidden units. So it does seem as though these individual differences are persistent really at any kind of neural system we're looking at, biological or artificial. And as you can imagine, perhaps what this is setting up is that if we have these differences in comparing within biological systems, and we similarly expect these differences in comparing within artificial systems, then if we want to start comparing across biological and artificial networks, as this really lovely figure by Dan Yeamans and colleagues suggests, we're just going to compound our problems. It's going to be really quite difficult to find a key way in which we can systematically compare between 2 systems that are themselves very diverse. And so the question I'm sort of posing in this talk is within a single data modality. So saying for the moment that we just want to cut down the noise and just look within biological systems or just look within artificial systems, how can we actually go about comparing learned representations across individuals and across experimental conditions? And what I've hoped, I've kind of motivated a little bit, is that this is not a trivial problem. This, I think, is really a core challenge for the fields of neuroscience and AI separately, and especially for us at this intersection of neuro-AI. So in my talk today, I'm going to talk about a few different strategies that are currently in place, and I'm going to, in particular, argue for alignment as a really exciting strategy that I think you'll hear a lot more about in the main talk as well. Main talks as well. And I'm going to lay out some considerations that you might want to have when you're working with alignment, and also provide some resources and recommendations for getting started with your own data, no matter what kind of data you're working with. So first, I want to lay out this idea of measuring dissimilarity with metrics. So this is perhaps the most common idea that you've likely already seen within the field. And the problem that we're trying to solve is basically this. We acknowledge that there are differences between individuals or across experimental conditions, and we want to effectively measure that dissimilarity, right? So we want a way to say, okay, this is how dissimilar we think these really are. This is an example that I've lifted from a talk by Alex Williams from his paper in NeurIPS last year. And basically what this is showing is if we have these 2 toy data sets, X and Y, if we have some metric, in this case, it's orthogonal Procrustes distance, it's going to provide us with a number. It's going to provide us with a quantification of how different these 2 particular toy data sets are for this given metric, right? And this can be really useful in a wide variety of circumstances. If you have 2 things that can't be compared for some reason, you can only look at second-level metrics. Maybe we want to just know these numbers. So this is really nice and useful. The problem, of course, is which metric to use, right? So the metric that you're going to use is in many cases constrained by the data that you have available, and quite often by your field-specific norms. So what do people expect you to report for this kind of data? Unfortunately, however, each metric is only going to provide one kind of insight into the underlying structure of learned representation. So there's no single metric that is best or is going to provide the most complete full insight into these data sets. Each one is going to provide a slightly different sense of the underlying structure and the differences between them. So for example, if we revisit these toy X and Y data sets, we saw with orthogonal Procrustes, which was going to just rotate X and Y to look more similar, that we get one number. But if we look instead at a canonical correlation analysis-based distance, which is now invariant to any linear transformation, we're going to see that we get a very different number, right? And it's going to say these are much more similar. And so it's not obvious, perhaps, which of these is right in any sense. But if we look at the field, what we see is that all of these measures and more are in really wide use. And so this is from a really lovely paper by Simon Kornblith and colleagues at ICML a couple of years ago. And what they're doing here is they're kind of sanity checking a number of metrics. So what we see is this is on the X and Y axis, it's two different instances trained of a single convolutional neural network, and they're calculating the layer-wise similarity using all of these different metrics. So a CCA-based metric again, and then some variants, this singular vector CCA, a linear regression, and then the metric they're proposing, centered kernel alignment. And what they argue is that for these two instances of the same network, so same architecture, same training data, different initializations, we expect that across these metrics, the diagonal would be bright. You expect layers to always look most like themselves across initializations, and the rest should be quite dark. But what we see is that for the given metrics, that's not necessarily the case. It very much depends on the particular metric of interest and on the given layers. And CKA perhaps performs best in this particular context, but by no means is it perfect, right? It's not only showing a bright diagonal and dark everywhere else. So it's capturing one aspect of the similarity here. And perhaps you say, okay, that seems really interesting, but the metrics that I'm familiar with have mostly come from the field. And perhaps the most common metric in the field, I would say right now, across neuro AI is representational similarity analysis, which has really been a workhorse for so much really exciting work that's happened across all of neuro AI. So for those who aren't familiar with representational similarity analysis, it's another metric. And the idea is that we're going to compare observed representational geometries. So basically what that realistically means is if we look at these two activation spaces in this schematic, what we're going to do is we're going to say for each of these four different stimuli, we're going to plot their relative activation across the observed voxels in this new activation space. And then rather than trying to make direct comparisons voxel to voxel, we're going to compare the distances between the different stimuli within these two activational spaces. So the distances are calculated within each activational space, and then the difference is compared across. And that allows us to derive a number, which we use to express the similarity, right? So in spirit, it's actually quite similar to all of the metrics I showed you previously, these CCA-based ones, the centered kernel alignment one from Kornblith and colleagues, and mathematically, they're actually very closely related as well. What's important, though, is that all of these measures, so CCA, CKA, RSA, are not true metrics in a mathematical sense, right? And this means that they don't satisfy a couple of important properties that we expect from metrics, one of which Alex Williams and colleagues pointed out just last year is the triangle inequality. So they don't meet kind of the Pythagoras intuition of how points on a triangle should be laid out if you have 3 points. And the problem with this is that, okay, it doesn't meet a mathematical definition, but in practice, it can significantly complicate downstream analyses. So if you're just deriving the numbers to have the numbers, totally fine. But if you want to use the numbers to do something else, if you want to use a CKA value to do a clustering, for example, or a statistical analysis, it can be really quite difficult to do that in a systematic way. So, all right. We have differences between systems, biological and artificial, and we have ways to quantify it. with metrics. And I don't want to understate the role that metrics have played in NeuroAI. They have provided hugely important insights into how systems are organized. And there's a lot of really active, compelling work ongoing to define more rigorous metrics that meet mathematical definitions and still work well with neuroscience and artificial network data. But at the end of the day, each metric is still only going to provide one perspective on the underlying representations that are learned in each of these systems, right? And so if we want to have not just metrics, but more than metrics, we're going to need to think about how we can actually compare across the systems themselves rather than only at the second level kind of extracted information. And to do that, I'm going to argue that we can think about alignment as a means to uncover richer, more dimensional, multidimensional information directly from the neural systems themselves. Okay, so yes, I see Pierre in the chat encouraging questions. Please don't hesitate to ask any questions. This is obviously a good point, but I'm very happy to be interrupted at any point. All right, so let's talk about alignment. So the basic premise of alignment is that whereas with metrics, effectively what we're doing is we're operating in the different spaces from different individuals or different experimental conditions. With alignment, what we're trying to do is directly bring that disjoint data into the same space. So we can make comparisons across them, so we can think about model fusion. There's all kinds of really exciting things that become possible when your data is actually comparable. So alignment is a pretty broad term, and there are lots and lots of methods that fall under this umbrella. So alignment can be done in a variety of ways. It can be done in high or low dimensional space, so you can work directly in the space of the data that you have, in voxel space, in sensor space, in the full hidden units of your layer space, or you can learn latent factors, a lower dimensional manifold, and work on aligning that instead. You can also do alignment using labeled or unlabeled experimental data. So you can do it knowing correspondence between your time points and leveraging that to help drive your alignment, or you can do it without knowing correspondence between your time point, where you're really just looking for these really rough heuristic comparisons across disjoint distributions. And importantly, depending, which we'll talk about in a little bit, but in many cases, these alignments can then be reused in new data, which opens up some really exciting possibilities when that's available. So this figure from Guntupalli and colleagues might look quite similar to the figure I showed you earlier for RSA, and it's indeed very much the same in spirit. So here, rather than those 4 labeled stimuli, we have 3 different labeled movie frames. We still have our 3 voxels, and we're looking at the relative activation of each voxel for the 3 movie frames. But importantly, unlike in RSA, where what we'd be doing is calculating, or most of the metrics I discussed, excuse me, where what we're doing is calculating the distance between these different stimuli, what we're going to do now is actually derive these transformations and then use those transformations to move our data into some new common representational space or common activation space. So this particular method that was proposed by Guntupalli, excuse me, and colleagues is known as hyperalignment and is based on Procrustes, which is that same method that I alluded to earlier. But there are many, many methods that work exactly like this in a high dimensional, full voxel, full sensor space to bring everything into alignment into one dimension where they can be directly compared. Of course, as I mentioned, it doesn't necessarily have to be done in high dimensional space. So this is another method, a schematic of another method that was originally proposed by Chen and colleagues and is really nicely summarized here by Cohen and colleagues. So this is called the shared response model. And what's happening here is just as before, you have some shared stimulus in this case. So this is labeled data where you know that everyone was watching the same movie, for example, let's say, if you're working with human data. And now what you're going to do is, rather than just looking directly at the voxels, you're going to find some joint low dimensional factorization where you have shared time components across individuals, and then every individual gets a spatial component as well. So you have this low dimensional K, which is less than the overall dimensionality of your data, and you're going to be able to have the spatial component for individuals and the time component shared. So you're learning importantly, what's really happening is you're learning some low dimensional factorization of your data that's stable and shared across individuals, right? So you don't just have to think about it in the high dimensional space if you want to look for latent factors. So, okay, this was 2 slides, and I threw multiple alignment methods at you. So as you can imagine, there are lots and lots of alignment methods that are out there that fit in these broad categories of high and low dimensional labeled and unlabeled data. And, whoa, got clicked on that. For an alignment method, it's not necessarily clear which method should be chosen right away, right? So much as for metrics, which alignment method to choose is data dependent. And in fact, at this point, it does remain largely guided by field norms. So to systematically explore the impact of alignment methods is something that is still really important within NeuroAI, I think. And as part of this, Thomas Bazet and myself, with our collaborators, went and tried to do a systematic benchmark of alignment methods in fMRI data. And just to provide a sense of how you can evaluate fMRI methods, I wanted to highlight just a few key results from this. And in particular, I wanted to highlight this framework we used, where we evaluated the success of alignment using intersubject decoding accuracy on unrelated downstream tasks. Right? So what we did here was we looked across the field with functional neuroimaging, and we found data sets that had a lot of data for each participant, and some of that we could use for alignment, and some of that we could use for a separate unrelated decoding task. So we had a range of different kinds of alignment data, covering movie watching, audio movie listening, task contrast maps, visual images, and a range of decoding categories, right? So visual working memory, music, images, sounds, language. This is really a broad span of what we'd usually look at in cognitive neuroscience. And so when we used alignment on these data, we actually found that we could recover a significant amount. So first, let me just explain this figure to you, and then I will walk you through what I mean by a significant amount. So here, what we're seeing is this is the intersubject decoding accuracy. And because, well, my pointer is the same color as the dot, so I'll move that away. Because we did intersubject decoding, each dot is a different cross-validation fold. So this is a different subject, and the subjects are colored according to the particular data set that they come from. And what we're comparing against the 0% line is if we didn't do any kind of functional alignment, if we just compared using anatomical alignment, assuming that if we moved everyone to the same anatomical space based on their brain landmarks, that we would be working in the same functional space. So we can see that these 2 methods, one of which I just showed you a minute ago, the shared response model, this low dimensional labeled data, and piecewise optimal transport, which is a high dimensional labeled data method. So we see that these are on average across participants about 5% improvement over anatomical alignment. And if we just did a within subject decoding minus intersubject decoding, so trying to see how much we lose when we move from the individual to the group, we'd see that we lose about 8%, 8.5% of decoding accuracy. So this gain of these performing methods is a little over half of what we normally lose to group variability. So this is pretty impressive in terms of gains. And also importantly, what we see, if we look at the signal itself, is that we see that we are not wildly distorting the signal, we're not losing signal. we're actually able to preserve the signal in a really structured way. So for example, if you look at piecewise optimal transport compared to the target individual we're aligning to, the group aligned using piecewise optimal transport is still picking up the peaks of the target individual's contrast. So we're really preserving signal specificity when we do this alignment. So that was one example of aligning in out of sample. So we derived our alignments on the movie, we applied them and held out decoding data. Depending on the particular kind of data you work with, that may not always be possible. So for example, a really big application of alignment actually is brain-computer interfaces. And here you need to find a way to on-sample or in-sample align because you don't necessarily have offline data that you can work with. So in this case, what we're trying to do is we're trying to calculate our alignment online and assess the performance as we go via decoder accuracy of participant movement or handwriting or whatever else our brain-computer interfaces is helping our participant to do. So as really nicely illustrated in this figure by Debagia and colleagues, what we're trying to do is to estimate a low-dimensional manifold. So again, this latent factor alignment, we're going to align it, and then we're going to take those transformations and apply it back out to our high-dimensional sensor or implant data. And what's really cool about this is that, talking to people who do a lot of BCI work here at Stanford, they'll talk about how every session where they have a participant, they have to have a recalibration session at the start. And this is really costly. This takes a lot of time. This, hampers participant engagement with the brain-computer interface. So alignment provides a really nice way to avoid constant decoder retraining for each experimental session, because otherwise, as I alluded to in the beginning, you can just see that you're losing accuracy over time. All right, perfect. Okay, so just looking at the chat, the slide deck will be shared. Yes, absolutely, that's a great question. So I will make sure that gets to you. And if there are any other questions, please don't hesitate to add them. Okay, so stepping back, we've talked about metrics as one means to look at dissimilarity. We've talked about alignment as a means to actually move data into the same space, and in particular, kind of the framework for alignment, where you have high and low dimensional data, labeled and unlabeled data, and how that can be used in a variety of settings, both training and then applying on unrelated data, and in the case of some settings where it's required, actually working online alignments within the same data. So this seems really promising. It seems like a really broad direction, but there are some, I think, major stumbling blocks that hit a lot of folks when they're considering working with alignment that I wanted to point out before we think about how we could actually get started with it in our own work. So when you're thinking about working with alignment, there are a few high-level concerns, I think, that arise. And I could think we could frame these as decision points, really. The first is choosing the right feature space in which to do the alignment. So it's all well and good to say that we'll align some data, but what does that data look like? If we can choose going in what kind of data to collect, how do we choose what kind of data to collect? The next is handling unbalanced alignments. So most of the methods I've discussed so far really assume that there's a one-to-one correspondence that you're going to be able to find in your alignment, and that's often not the case. We don't always expect information to be perfectly preserved between different brains or between different neural network instances, for example, if the layers are different sizes. And then finally, we may need to think about considering template-based alignments. So rather than aligning to data that we have, that we know exists, if we have many, many sessions, many, many instances or participants, we may need to think about how can we align not one-to-one, but to some shared or common template. So first, choosing the right feature space. And I think this, may not be as obvious when you first start, but I think it's really quite critical, is that when applying low dimensional alignments, the selected features subspace will significantly impact the results. So this is a supplemental figure from that study I just showed you, where we used piecewise SRM and optimal transport to look at intersubject decoding accuracy. But really what I want to hammer home here is this is a grid search for the hyperparameters for piecewise SRM. So you can see K, which is the hyperparameter for the dimensionality. You can see this other parameter where we look at the number of parcels, and you can see that the range of values is really quite wide, right? So what we're seeing is that the specific hyperparameters that we're using to select our feature subspace are going to significantly impact our results. And it is something that really needs to be carefully considered, particularly in this case for subspaces when you're working with low dimensional alignments. So for example, if you're thinking in a BCI application, what are you going to use to learn your manifold? What particular algorithm are you going to use there? That can have a really high impact downstream. Another point is that, if you're going about and you're collecting data, so if you're designing a training set or you're designing a stimulus set for participants, if you're trying to do an out of sample alignment in particular, it may not be obvious how best to match the characteristics of your training data where you learn the alignment with where you're going to apply it. And this is really complex, I would say. This is something we're still trying to understand, but we do have some broad rules of thumb. So for example, if you're going to apply to something that's a language task, what you learn your alignment in should probably include language in some form, right? It shouldn't be visual only clips. So if you can carry information over, it will make the distribution alignment a bit easier later, so long as you're covering similar kinds of information, both where you're learning and where you're applying your alignment. Another important point is unbalanced alignments. So this is a really nice figure from Twal and colleagues from this year at NeurIPS. So in some cases, in fact, in many cases, depending on your particular data modality and question, your source and target data may not have a one-to-one matching. So in this case, for example, we have two participants. We're looking at a contrast map for the same contrast, like an auditory language contrast. And what you can see is that for each participant, it's generally within the same set of brain regions, but we don't see that it covers the same spatial extent, right? And so if we were to enforce that all information from subject 7 moves to subject 9, we might, or vice versa, we might see some kind of odd or undesirable effects. And instead, what we can do is actually allow for an unbalanced alignment, allow for not all of our information to be matched. And this is really useful quite broadly in this example, for example, where different individuals have differently sized functional areas in artificial neural networks, where you have layers, which maybe you have the same number of layers, but within a layer, two architectures have very different numbers of hidden units, allowing things to decay when you move them during the alignment can really help to make sure that information is actually preserved. And this means that unmatched information is going to be discarded rather than complicating your alignment and making it harder to understand what's happened after the fact. So this is a really nice example from Singh and Jaggi at a NeurIPS paper a couple of years ago. What they have here is they have a specialist network. So this specialist network had access to a different data class than the generalist network. And they see that if they initialize them differently, much as we've seen generally where you have different initializations, if they just average them, they see that with vanilla averaging, this blue line, they see that the performance really falls off. So this is just averaging all of the model weights between the two different models. Whereas if they use this kind of alignment, this optimal transport averaging that they argue for, they see that they can really conserve the performance. And when they initialize them the same way, they see the effect isn't as dramatic, but it's still present. So this optimal transport averaging, rather than just kind of a naive averaging, performing some kind of alignment first and then averaging, allows you to really recover performance between models that otherwise, because of individual differences, would be very difficult to compare directly. Finally, the last kind of consideration that I wanted to make here is about functional templates for alignment. So in all of the cases that we've considered thus far, we assume that we're performing alignment to a real known target. So this would be, for example, a previous experimental session or an existing model or participant. But in many cases, it would be preferable to align to a functional template. So for those of us in neuroscience, this might be analogous to the idea of anatomical templates, such as those provided in Montreal at the MNI. Now, this is really appealing and really quite difficult. So calculating such a template is an active research area. And initial methods have come out proposing various solutions. So for example, Haxby and colleagues have proposed developing templates using generalized Procrustes, again, referencing that Procrustes method I mentioned earlier, to an inferred average, or more recently, that figure from Twell and colleagues is from a paper where they also talk about using Wasserstein-Berry centers to develop an average. There's no good answer there. There's no good answer there as to what the correct template would be or should be. So for now, I would say this is definitely something that folks are working on, and I'm sure more help would certainly be useful. But this is a very active area. Okay, perfect. So looking at the time, let's say that you would like to get started with alignment. How could you go about getting started? First, a lot of people have very kindly shared code for different ways to do this using different kinds of data types, right? So several of these are designed primarily for fMRI data, in particular BrainIAC, fMRIline, and PyMVPA. But Neuroline here, from Eva Dyer and colleagues, is actually designed for data from systems neuroscience, so like calcium imaging data, spike sorted data. OT Fusion from Singh and colleagues is designed for model fusion with neural networks. And this allows you to, again, align multiple models and then perform an average of the parameters that conserves information rather than discarding or losing information between the individual differences of the different models. So code is great, but it's also useful to have a general framework in mind for how to get started. So if we have these resources, if we have the data, and we're convinced that alignment is something we should be thinking about across all of these different systems, how can we go about getting started? I would just provide some really general recommendations, one of which I think is critical, which is consider how you will evaluate the success of alignment on your data. So for example, in the paper I talked about from Thomas Bazet and myself and colleagues, what we found was that it's non-obvious in many cases how to actually evaluate alignment success. There we used intersubject decoding. There are a variety of metrics that you could use to evaluate success, but I think in many cases people will assume that, okay, if I calculate and then apply an alignment, then I've improved similarity and I can now perform whatever analysis I want. And it's really hard to know if that's true, right? I think we're still in very early days of learning what kinds of alignment are best for particular data types and for particular questions. So for now, I would really encourage you to consider evaluating the framework by which you will evaluate the success of alignment before you start applying it to your data. I would also say just look at the literature in your field. So alignment is definitely gaining steam right now. I feel like I'm seeing more and more all the time different groups and papers that are using different kinds of alignment methods in their own data. And so I would say take a look in your field and see what alignment methods have already been applied. So if you're working in brain-computer interfaces, there are some emerging best practices there. If you're working in fMRI, if you're working in calcium imaging, all of this can really help you to get a sense of what alignment methods are going to be useful for your data type. But of course, you'll still need to evaluate, was the application successful for your particular question? And then finally, I think this is something that's emerged more recently and is incredibly important, is evaluating if your application is likely to have unbalanced information between source and target data sets. So if you're working with information where you do think that you're not going to have a one-to-one correspondence, or if you think there's even a chance that you might not have a one-to-one correspondence, choosing alignment methods that can handle this kind of unbalancing is really important. And that's something you can test in your data. You can say, once you have this framework by which you're going to evaluate success, you can say, okay, if I allow for unbalancing versus not, how does that change the success of my alignment? What do I differentially learn? All right. So with that, I want to thank my supervisors, Russ Poldrak and Scott Linderman, the WUSI Neuroscience Institute, the labs, and obviously Unique and McGill, who've been so instrumental in all of this work that I presented here. And I want to leave you with a few take-home ideas about alignment for our field in neuro AI and some exciting, I think, future directions. So thank you.